Hello everyone. So here is our recording. I'm not quite sure how to do the videos. Everything I did was not showing up, so I'm going back to the PowerPoint and doing a talk over. We're talking about the heart. Some statistics is at a rate of 75 contractions per minute. How many times is our heart beating a minute? Average is 75. So at this rate, a human heart would contract approximately 108,000 times a day. And that's more than 39 million times in 1 year and nearly 3 billion in a 75-year. So we know that we have, on average, 5.25 liters of fluid per minute that's perfusing through our body. We have through our body perfusing or contracting would be 14,000 liters per day, and that would be the equivalent over 10 million liters or 2.6 million gallons of blood being sent through our body, through all these vessels, whether it's arteries, capillaries, veins, through our body. So the general function we already said of the cardiovascular system is simply to pump blood throughout our body so that this is going to help our cells that are doing work to meet those needs. Remember, our body needs oxygen. That oxygen is going to be important in making that ATP so that our cells can do work. So it says to remain healthy, all cells are going to require a continuous delivery of oxygen and nutrients. Also in our blood, it's not just oxygen, but we're getting these nutrients. Also that even though the blood is bringing oxygen and nutrients to the cell, it's also removing waste from our cells, which is our carbon dioxide and then other waste products that are occurring within the cell. So all of our body systems depend on our cardiovascular system to meet those needs. Again, that heart is nothing but a muscle, and it's pumping this blood throughout our body. So as you can see here, we've got our heart, we've got our artery, veins, those capillaries. So these are all going to be those components system that's going to be responsible for that movement of blood, or how the blood is moving through the body, or the containers in which they're moving, if you want to think about it that way. So the cardiovascular system, when we talk about perfusion, so perfusion is just simply that delivery of blood to the tissue or in the tissue. So again, perfusion is to help maintain the health of all of our body cells. So maintaining adequate perfusion requires both the pumping action of the heart and healthy patent, and that means these vessels in which this blood is flowing through has to be open and unblocked. So if we hear the term patent airway or patent blood vessels, this just simply means that they're open, there's no occlusion there to stop that flow of whatever is going through it. So I want you to ask yourself this question: what would happen to tissues if the heart fails to pump or the vessels become hardened or occluded? So let's look at the components of this. So we're going to have different components of our heart. So we've got our blood vessels. So our blood vessels are going to be our arteries, our capillaries, our veins. So our arteries are what's going to be transporting blood away from the heart. So arteries, however, we like to think that when it's carrying blood away from the heart, there's always going to be oxygenated blood. However, that is not the case. Arteries do not always carry oxygenated blood. We have veins that are transporting that blood to the heart, and we like to think that veins are carrying that blood that doesn't have the oxygen in it. That's not always the case. We're going to see where we have arteries and veins that are doing the complete opposite. So arteries carry blood away from the heart, veins carry blood to the heart, capillaries are where we're going to see that exchange of gases. So in the lungs, it's going to be that exchange of gases of oxygen or carbon dioxide for oxygen. We know that the heart is going to be the center of the cardiovascular system. And when we look at the heart, when we look at that structure of the heart, we know it's a hollow or hollow four-chambered organ that's going to pump blood throughout the body. So when we start looking at those anatomical features, we know it's going to have two sides, a left and a right. It's going to have greater vessels attached to the heart, and we're going to have two sets of valves located within the heart. So I would draw a heart, and since I know these things about it, these anatomical structures, then I'm going to now start being able to categorize what this looks like. You have this in your books. We've already said that. We have these two sides. We have on the right side, we've got our right atrium. So I want you to remember, atrium is a receiving chamber where ventricles are going to be what's pumping. Those are going to be those chambers that's pumping the blood away. So if you hear the term atrium, well, I know blood is being collected there. If I hear the term ventricle, I know that is where that blood is being pushed or pumped out of. So on both sides, on the left side, we've got a right atrium and a right ventricle. When on the left side, I have a left atrium and a left ventricle. Again, go back to that those anatomical structure. So then with our greater vessels, we have our arteries and our veins. So those arteries, we've already said, is going to be pumping that blood or transporting that blood away from the heart. Here is where we have our pulmonary trunk. So that pulmonary trunk is going to pump or is going to transport blood from that right ventricle, and then our aorta is going to transport blood from the left ventricle. So you've got these greater vessels, so remember that. Our veins is going to be transporting blood toward the heart. We've already said that. So we have our vena cava. So we have superior vena cava, inferior vena cava. So if you look at superior vena cava, it's going to be superior at the top. Inferior is going to be, inferior means below, so that's going to be found at the bottom. Again, remembering these anatomical terms. So your vena cavas are going to, that blood is going to go into the right atrium. It's going to drain blood into the right atrium. And our pulmonary veins are going to drain blood into the left atrium. So these veins, remember, they're pushing towards the heart. And then we have our valves. So we have two valves. We have our atrioventricular valve, or our AV valve. And this is going to be, so our right AV valve and the left AV valve are going to be found between the atriums and the ventricles. So there's going to be that space between the atrium and the ventricle where we're going to find our AV valve. Our semilunar valve are going to be found where we have our pulmonary semilunar valve and our aortic semilunar valve. So just think about that. I'm not going to find my pulmonary semilunar valve in between my aorta and my left ventricle. I'm going to find that. So these are going to be found. So that's going to be found between my pulmonary trunk and my right ventricle. So think about where these valves, what it's saying, pulmonary semilunar valve and your aortic semilunar valve. So these are going to be those vessels. Here you've been given this, so you should be. So again, we have chambers of the heart. We have a receiving chamber, and then we have a pumping chamber. Remember, your atriums are going to be your receiving chambers, and your ventricles are going to be your pumping, pushing that blood away. And we're going to have different vessels associated with the heart. We've already talked about our superior vena cava and our inferior vena cava. We've talked about the semilunar valves. They're not on here, but we have our carotid arteries. We have our jugular veins. So we have all of these different major vessels that are heart. Again, those valves. So a valve is just simply to keep blood from flowing back into the heart. That's all those valves are for. It's like it's stopping. Once that blood goes through, those valves close, and that blood that passed through cannot flow back. So we have our pulmonary we have our pulmonary valve, and then we have our aortic valve. And then it's not going to show, it's not showing the semilunar valve. It's not showing. So how does blood flow in the heart? So we have our right atrium. Again, we said those atriums are those receiving chambers. So that right atrium is going to receive that deoxygenated blood from the body via your superior vena cava and the inferior vena cava. Both of these are pushing that blood to the right atrium, to that receiving chamber. So that blood is going to pass through the tricuspid valve into the right ventricle. Then the right ventricle is going to pump, remember ventricle's that pumping chamber, is going to pump the blood through that pulmonary semilunar valve into that pulmonary trunk. So then that pulmonary trunk is going to divide into your right and left pulmonary arteries. So remember, trunk now divides into two different arteries, which are going to carry that blood to the lungs. So remember, pulmonary means lungs. So if it's the pulmonary trunk, pulmonary arteries, this is going to the lungs, and this is where that blood is going to become oxygenated. Remember, our capillaries, those capillaries in our lungs where that oxygen exchange takes place, is going to be called your alveoli. So these alveoli are these little tiny sacs, and it's with the it's these tiny sacs that's going to be found in the um, in the lungs. So this is where we're getting that gas exchange. So we're exchanging the carbon dioxide that's in the lungs for now this oxygen being pulled out of these capillaries. So again, this is where that, and then the oxygen goes into the lungs, then it goes into, and that carbon dioxide goes back into those capillaries, and we've got this oxygen exchange. And again, when we get into more to that. So now that we've had this exchange, now the left atrium is going to receive the oxygenated blood from the lungs via the left and right pulmonary veins. So remember, going to the lungs was your pulmonary trunk divided into your pulmonary arteries. Now that oxygenated blood that's leaving the lungs is leaving via your right and left pulmonary veins. So again, those veins, what's leaving the lungs? It's going to be anything pulmonary. So remember, pulmonary is associated with the lungs. So that blood is going to pass through your bicuspid valve, or what we call mitral valve, into the left ventricle. So the left ventricle is going to pump that blood through the aortic semilunar valve into, so remember, aortic semilunar valve is going to be associated with your aorta. And then you have your systemic arteries branch off of the aorta and carry blood to the tissues where it becomes deoxygenated, because those tissues are pulling that oxygen out. So now it's getting what it needs. So now that blood is now deoxygenated. So now systemic veins are going to return the deoxygenated blood to the right side of the heart. So this is your blood flow, and you will need to. So again, we say we have two sides of the heart. We've got a left and a right. We've got different blood vessels that are going to be associated with both sides of the heart. So we say we have a pulmonary circuit and we have a systemic circuit. So when it's dealing with pulmonary, you already know that pulmonary means lungs. So this is going to include that movement of blood to and from the lungs for gas exchange, or what we say, that external respiration. And then we have our systemic system. So when you think about pulmonary, we're thinking lungs, we're thinking about gas exchange. Systemic circuit is where we're including that movement of blood to and from body tissues for gas exchange, internal respiration. So pulmonary circuit, when we're, again, it's external respiration. Systemic, you can think of it, where we're having that gas exchange for internal respiration. So again, here it's broken down into systemic and into pulmonary. See both sides, pulmonary circuit, and then we have systemic circulation. So where is the heart? And so the heart we know is going to be within the mediastinum, and it's going to be covered by a membrane called your pericardium or pericardial sac, and that cavity in which it sits is called your pericardial cavity. So here we've got that. We know we got the lung sitting on both sides. Again, that heart is going to sit in what we call that mediastinum, and it's going to sit within the pericardial cavity and be covered by your pericardial membrane. Again, we've got that fluid. Again, parietal is going to mean that membrane along the wall. Visceral is going to mean anything, that membrane that's covering the organ. We've got our pericardial fluid that's going to be located between your parietal pericardium and your visceral pericardium. Serous fluid. So what is pericarditis? Well, we know itis simply means infection. We know pericardium is going to be that membrane that's covering the heart. So this is going to be an inflammation of that membrane, and this is can be caused by bacteria, viruses, or fungi. And this is going to be associated with increased permeability of the capillaries within the pericardium. And this is going to result in that excess fluid accumulating in that cavity. And this can cause the heart to be unable to pump that blood, which can lead to cardiac tamponade and possible heart failure or death. Because now the heart can't beat, it can't pump that blood, because every time our heart beats, it's simply just pumping the blood. So it's going to impede that movement, and now our heart, we go into heart failure. We know our heart is more slightly rotated. Its right side is located more anteriorly, and the left side is located more posteriorly. So it's not like it's just when we look at the heart, it's kind of tilted to where the heart right side is more to the front and the left side is more to the back. And we say that the posterior superior surface of the heart is called the base, and then the inferior end is called the apex. We like to think that the base is the bottom. In this case here with the heart, it's going to be. So from the anterior view, we said anterior means front. The right atrium and right ventricle are going to be prominent. Remember, we said that slight rotation is going to cause the right side of the heart to be more forward or more anterior. And the right auricle is like a flap that's at the flap-like extension of the right atrium. It almost looks like we say it's the ear. That's what auricle means, is ear. The pulmonary, we have the pulmonary trunk. Again, that's going to divide into those left and right pulmonary arteries. And then we have our aorta, which is, we have our ascending aorta, then becoming our aortic arch, and then we have our descending aorta. Again, here we have, so we see in the picture, the posterior view. This is where we're going to see more of the left atrium, and the left ventricle are going to be more prominent. That left atrium is going to primarily form the base on the posterior superior surface of the heart. And this is where we're going to see those attachment points for the superior and inferior vena cava to the right atrium. And then we're going to have the pulmonary veins to the left of the atrium, because this is what's going to be carrying blood from the lungs. So then we have our coronary sulcus. Remember, when we were talking about the brain, we had their sulcus and gyri. So sulcus are just going to be those deep grooves. So the same thing, same concept, only now we're applying it to the heart. So that coronary sulcus is going to be those deep grooves that are separating the atria from the ventricles. Remember, we got our atrium and ventricles. So our interventricular sulcus, this is going to be a groove between our ventricles that extends from the coronary sulcus to the apex. Remember, we said the apex is going to be that tip at the bottom. So here we've got the coronary sulcus. Again, that's going to be separating the atria from the ventricles. So this is where we're going to find our right coronary artery, that small cardiac vein, the coronary sinus, and the circumflex artery. So this is where we're going to find all of these. Again, this is a better view of the heart. We've got our left auricle and our right auricle. It's kind of those, looks like little ears on both sides. We've got our superior vena cava and our inferior vena cava. We can see here. Again, this is all going to be pumping that blood into the right. So we have our epicardium, we have layers of the heart. We have our epicardium, our myocardium, and our endocardium. So each one of these is going to, we're going to be able to see. So remember, epi means outermost layer. Then we have myo, which means middle, and then endo. It's going to be that innermost layer. So the epicardium is going to be that outer layer of the heart. Myocardium is going to be composed of the heart muscles. And then we have our endocardium, which is going to be continuous with the endothelium, which lines the blood vessel. So just remember, epi, outside, myo, the middle layer, endo, the innermost layer. So we know that our right and left atrium are going to be separated by an interaurial septum. So this is going to be, so the right atrium, we said, is that receiving chamber for the systemic circuit. Then we have, so in that we have our tricuspid valve. Then we have our left atrium, which is going to be the receiving chamber for the pulmonary circuit. This is where we have our bicuspid valve or mitral valve. So both of these are going to have muscular ridges called pectinate muscles. You're right. So again, we said that the right and left ventricles are going to be separated by our interventricular septum. So both of these ventricles are going to have large, smooth, irregular muscle ridges called the trabeculae carneae. If it's in red, just remember this is what you're going to need to know. So your papillary muscles are going to attach to the chordae tendineae, which are going to be attached to your atrioventricular valve. This is going to be attached to both your tricuspid valve and your bicuspid valve. And when we look at them, they're going to look like little cords or strings. And this is what's going to help in the contraction of the heart. So these valves, again, we said that those valves are a way to ensure that that blood flow is flowing in one direction. It ensures that one way. We're like on a one-way street here. So with our atrioventricular valves, we have, again, our tricuspid valve, our bicuspid valve. Then we have our semilunar valves, which is going to be our pulmonary semilunar valve and our aortic semilunar valve. So you can read this about teenage athletes and sudden cardiac death. So again, cardiomegaly, this is just where, remember, the heart is a muscle and that the walls of the muscle can get really thick. And a lot of times when we have in young athletes, this is where that, so as the heart is enlarging, it's creating an environment that these blood vessels are now narrowing, and that blood flow cannot get through there. And a lot of times these deaths this is a heart defect, and sometimes a lot of times they don't know the death has occurred. When we listen to our heart sounds, we can have abnormal heart sounds called a heart murmur. And again, this is going to be an indication, or first indication, that there is a heart valve problem. So remember our tricuspid valve, we have bicuspid, or those semilunar valves. So there could be an issue with those. So functions, we're going to know that we have fibrous skeleton of the heart tissue. So again, this is going to provide structural support of the boundary between the atria and the ventricles. It's going to provide a rigid framework for the attachment of the cardiac muscle tissues. Again, it's going to act as an electrical insulator. So remember, we talked about those action potentials. This is going to prevent the propagation of action potentials directly from the atrium to the ventricle. So this is going to prevent, we don't want them both contracting at the same time. One has to contract, then the other one can contract. So this is going to prevent the right atrium from contracting at the same time that the right ventricle is contracting. So we know that our myocardium, remember the myocardium is going to be that middle tissue, is composed of cardiac muscle tissue. And again, it's going to be relatively short branch cells that are usually house one or more nuclei. You're going to have the sarcoplasm, which is called the sarcolemma. You have your sarcoplasmic reticulum. Again, remember, in muscle contraction, this is where we have the calcium is released so that the action potential can take place. And then we have our myofilaments. So in our myofilaments, what's different about cardiac muscle versus skeletal muscle? They're both striated. However, in our myofilaments, we're going to find these intercalated discs and those cell-to-cell junctions. And this is what causes electrical rhythm. So we know that the heart is going to need a lot of energy, right? It's going to need a lot of ATP because our heart is constantly beating. We already saw at the beginning how many times our heart beats in a day versus over a period of time. So again, it's going to need extensive blood supply. It's going to need more mitochondria. And then we have our myoglobin. So our myoglobin proteins are binding to oxygen when that muscle was at rest. We have that creatine kinase. So this is going to also be that enzyme that's going to catalyze that transfer of that phosphate that needs to bind to another ADP so that we can have that ATP form. And when that creatine kinase forms, then we get the formation of ATP and creatine. So cardiac muscle cells rely mostly on aerobic cellular respiration. So cardiac muscle is quite susceptible for failure if akismic, I know I'm probably not pronouncing that right, which just means low oxygen conditions prevail. So in an environment of where there's low oxygen, our cardiac muscles are not going to be working. So any change that interferes with blood flow to the heart can cause damage or death to the cardiac muscles. So here you've got some fill-in. So if you'll go in and fill those in. So the right and left coronary arteries are positioned within the coronary sulcus. Remember, it's just those grooves and it supplies blood to the heart. So these are coronary arteries. So our body tissues are generally served by one artery called an end artery. So coronary arteries are functional end arteries. So if either the right or the left coronary arteries become blocked, then that blood flow becomes insufficient, and then this can result in what we call a myocardial infarction or heart attack. So you want to use the acronym RAMP to help you remember the three major coronary arteries that come off the right side of your heart. So R is going to, the right coronary splits into the right marginal artery and posterior intraventricular artery. Then for the left side, you're going to remember LAC, the left coronary artery, anterior and ventricular artery, and the circumflex artery. So right side is RAMP and left side is LAC. So again, here is another. So our coronary veins. So these are going to transport deoxygenated blood from the myocardium through one of several cardiac veins. So that's what our cardiac veins are going to do. They're going to be transporting that deoxygenated blood from the myocardium. So we have our greater cardiac vein, and this is going to be located in your anterior interventricular sulcus alongside of the anterior intraventricular artery. Then we have our middle cardiac vein, our small cardiac vein, but we have a coronary sinus. So that coronary sinus is going to be that large vein that lies within the posterior aspect of the coronary sulcus. So this is going to return blood from the coronary circulation to that. So we said a myocardial infarction is going to be called a heart attack, and it can be potentially lethal, or meaning it could cause death. That results from sudden and complete occlusion of a coronary artery. So that means that coronary artery is blocked by a substance called plaque. Usually in men, they have right arm pain. Doesn't always that the heart hurts, but we have what we call referred pain, and depending upon where that referred pain is, it can start in the jaw. I know for women, it can look different. Sometimes so the heart's conducting system. So the heart has special cardiac muscle cells located internally into the endocardium. These cells don't contract, they just conduct electrical. So these are going to be our SA node. We call this the pacemaker of the heart. You have your atrioventricular or AV node, and this can take over if your SA node is not properly functioning. Then you have your AV bundle, or what we call the bundle of His, and this is going to divide into the left and right bundle branches. And then we have our Purkinje fibers, and these are going to begin at the apex of the heart to the walls of the vein. Again, we have our SA node. Again, that's the pacemaker of the heart. We have our AV node. So this is like, and then we have our AV bundle, or the bundle of His, and then we have our Purkinje fibers. So it's asking you here, which is the correct sequence of events in the transmission of an impulse through the heart muscle? So put this in order as to the way they're supposed to go. So innervation of the heart. So both your heart rate and strength of contractions are regulated by your cardiac center of the cardiovascular center within the medulla oblongata. So again, we've talked about parasympathetic and sympathetic when we're talking about hormones. Now we're going to talk about how does this innervate into the heart. So our parasympathetic innervation, we know anything that's parasympathetic system is going to decrease while sympathetic is going to increase. So with our parasympathetic innervation, this is going to send signals via the vagus nerve, which is going to result in, which is your cranial nerve 10, which is going to result in a decreased heart rate. Then we have our sympathetic innervation, which is going to send nerve signals along the cardiac nerves, which is going to increase both your heart rate and the force of contraction. So it's like pumping that blood out. So now we're going to talk about the stimulation of the heart. So the physiological processes that are associated with heart contraction are organized into two major events. We have our conduction system, and then we have our cardiac muscle cells. So that conduction system is where that electrical activity is being initiated at that SA node. Remember, that's your pacemaker of the heart. And again, this is where that action potential is going to be transmitted through, and that the action potential, is going to be transmitted through this conduction system. Remember, action potential is something is happening. Conduction, something is happening. Then you have your cardiac muscle cells. So as that action potential is spreading across those cardiac muscle cells, those sarcomeres within the cardiac muscle cells contract. So this event occurs twice in cardiac muscle cells during a heartbeat. So when we talk about action potentials, we have that resting membrane potential. So it says nodal cells at rest. So nodal cells have an unstable resting membrane potential of negative 60 millivolts. So again, that voltage-gated sodium channels are located in your plasma membrane of the nodal cells, open in response to that change in membrane potential. Remember, these channels and leaks, they respond to any kind of change that's occurring in that plasma membrane. Then we have autorhythmicity. So again, that SA nodal cells are going to be unique in that they exhibit autorhythmicity or automaticity. That means it's doing this on its own. It's automatic. So this means that they are capable of depolarizing and initiating an action potential spontaneously without any external influence. So remember, for an action potential to take place, we had to have that change in that plasma membrane. That resting membrane potential had to change. Now with this, your SA nodal cells, they are capable of depolarizing and initiating that action potential without there actually being a change in that resting membrane potential. So again, we're talking about action potentials. We have that the voltage-gated sodium channels were opening. There's been some kind of change. Or in the case of this, events at the SA node, we know that does not have to have any changes occurring for the SA node to initiate that action potential. So again, we're going from 60 to 40 millivolts, which is that threshold value. Then we have depolarization of that membrane. So that changing of that membrane potential now triggers those calcium entry into those nodal cells, which causes a change in that membrane potential from negative 40 to just above 0 millivolts. Now we have our repolarization. Now we're going back to that resting membrane potential, or what that membrane potential was. So now those calcium channels are going to close, and now this is where your potassium channels are opening up. So now potassium flows out to change the membrane potential back to negative 60 millivolts. So this process normally takes about 0.8 seconds. At rest, this is what results in a heart rate of 70 beats per minute. So this is what allows for that resting heartbeat. So your normal resting heartbeat of 75 beats per minute is due to that continuous parasympathetic stimulation of the SA node by the vagus node. So this slowing of the heart rate is called our vagal tone. So again, our resting heartbeat. So you go to the doctor, they're checking your resting heartbeat. Now you know that it's this going from polarized to repolarization in our nodal cells that is allowing for this to occur. So it's due to that continuous parasympathetic stimulation of your SA node by the vagus nerve. So pacemaker potential. So we know that our SA node is what we call the pacemaker cells. So the ability of these cells is to be able to reach threshold without there being any stimulation. We don't need there to be stimulation for this action potential to take place to have a resting heartbeat. So our heart contractions do not require stimulation by our autonomic nervous system. So action potential we know is distributed through both right atrium and left atrium. Those atriums are going to depolarize and then they're going to contract. Then that action potential is now relayed to your AV node. That AV node now is going to cause a delay in the actual potential. And now that action, so this is going to allow those atriums. Remember, those atriums are going to be those receiving chambers. We've got blood in these receiving chambers. So this is going to, that delay is going to, of that action potential, because remember that action potential is causing these, each one of these to contract. So this is going to allow enough delay for the atrium to finish contracting, to force that blood or to push that blood into the ventricles so that there is complete ventricular filling. We don't want any blood left over in our atriums. We want that contract or action potential to be delayed enough so that that blood can go in from the right atrium into the right ventricle. So these action potentials are going to travel from your AV node through your AV bundle to your AV bundle is also called the bundle of His, if you ever see that in books or anything I give you, to your Purkinje fibers. Those action potentials are traveling both through your ventricles via gap junctions. So this allows for both the ventricles to depolarize and contract at the same time. Remember, in our atriums, it's just depolarizing and contracting. But however, this allows for both ventricles to contract at the same time because these action potentials are traveling through gap junctions. So a pacemaker other than your SA node is called an ectopic pacemaker. Excuse me. So this is where our AV node comes in. If that SA node is not working properly or is malfunctioning, then our AV node can become that default pacemaker. And then that establishes the rhythm of the heart as well. So survival is possible because a rhythm of 40 to 50 beats per minute pumps sufficient blood to sustain life. So if both your SA node and the AV nodes are not working properly or they're not functioning, then that slows the heart rate down to 20 to 40 beats per minute. And then this is when we have to have a mechanical pacemaker put in place. This is just the action potential. So cardiac muscle cells, unlike our skeletal muscle cells, cannot exhibit tetany. So this distinction is critical for the heart to be able to function as a mechanical pump because it's moving blood. So that it does not exhibit tetany. So that refractory period is when the muscle cannot be re-stimulated to contract. So remember, tetany is just involuntary muscle contractions. Like whenever you have muscle spasms, it's tetany. So again, this is going to, the cardiac muscles do not exhibit this. So what is a cardiac cycle? So all events in the heart from the start of one heartbeat to the next is what we call a cardiac cycle. So this is going to involve systole, which is the contraction of the heart, and diastole, or diastole, which is the relaxation of the heart. I can't talk today. So systole is contraction. Diastole is the relaxation of the heart. So as that cardiac cycle begins, all four chambers are at rest. So blood continues to return to the right atrium and to the left atrium. Remember, these are our receiving chambers. So passive filling of the ventricles is occurring. So everything's happening simultaneously, if you want to think about it like that. Only we don't have both sides of the heart, or both your right atrium and right atrium and your right ventricle contracting at the same time. So we have passive fillings of the ventricles is occurring. You've got your AV valves are open and your semilunar valves are closed. So that atrial contraction is initiated by the SA node. Remember, that's our pacemaker cell. So contraction of these atrial valves removes the remainder of the blood that was within each atrium into the right or left ventricle. So ventricular filling is completed until termination of the atrial contraction, and the ventricles now hold their maximum blood flow. So this is going to end that diastolic volume, or EDV, and then the atrials can relax, or atriums can relax and remain relaxed until the beginning of a new. Then we have the ventricular ejection. So remember, our ventricles are what's pumping that blood out. So that movement of the blood from the ventricles into your arterial trunks are going to occur during this phase of contraction. So the pressure we said are increasing within those ventricles, and it's going to exceed the pressure within those arterial trunks. So your semilunar valves are forced open, and now your AV valves are going to be closed. So the amount of blood that's pumped out during a ventricular contraction is termed your stroke volume. The blood remaining in a ventricle at the end of systole is called your end systolic volume, or your ESV. So to find my ESV, I'm going to subtract my EDV minus my SV. So this is I'm taking my diastolic and subtracting my stroke volume from it to get my ESV. Sorry. So ventricular balance. So this is going to where I've got equal amounts of blood are normally pumped by two ventricles, our right and our left ventricle. So sustained pumping of unequal amounts of blood may result in edema, which is that excess fluid that's being found in that interstitial space. So the right ventricle has to pump the blood to the lungs, which is a shorter distance, and it doesn't require as much pressure because it's just going to the lungs. Just think it's right ventricle, now it's pumping blood to the lungs. And we already know where the lungs are on both sides of the heart. However, this is why the left side of the heart requires more pressure, because it has to pump blood at a greater distance. So it's going to require that higher pressure. So it says the left ventricle has to pump blood throughout the systemic system, our whole body. So again, this is a longer distance and it's going to require higher pressure. So this causes the left ventricle to be larger and stronger than the right ventricle. So when we look at the left, it is larger. Here's another question for you. Cardiac output is defined as the amount of blood that is pumped by a single ventricle, left or right, in 1 minute, and is typically expressed as liters per minute. So cardiac output is determined by the heart rate and stroke volume. So to find cardiac heart rate is, to find cardiac output, I'm going to take my heart rate and I'm going to multiply it by the stroke volume. So it says at rest, heart rate is about 70 beats per minute and stroke volume is about 70 milliliters per minute. What is the cardiac output? So what percentage of blood is pumped every minute? So figure those. Here is another information for you to fill out. So what is cardiac reserve? So cardiac reserve is an increase in cardiac output above its level at rest. So is this a measure? This is a measurement of the level and duration of physical effort in which an individual can engage. So cardiac output may be increased by about 4-fold in a healthy, non-athletic individual, and up to 70-fold in a highly trained athlete. So individuals with weakened heart may have a little cardiac reserve and will not be able to exert themselves. So heart rate can be altered by external factors that act on both the SA node and the AV node. So we have what we call positive chronotropic agents that can cause increase in our heart rate. So this is a part of that sympathetic division. Remember, sympathetic increases our heart rate. So this can be a neurotransmitter, norepinephrine, and that causes that. We have causes of release of norepinephrine and epinephrine, which are going to bind at those beta-1 adrenergic receptors of the heart. An influx of calcium ions, thyroid hormone. Too much of that thyroid hormone being released can cause an increase in heart rate. Nicotine increases the release of norepinephrine. Cocaine inhibits norepinephrine, that reuptake of it. So if it's sitting there, cocaine inhibits that. Caffeine inhibits the breakdown of cAMP. So this is why too much caffeine can be fatal in some cases. So all these energy drinks that have all this caffeine in it can lead to heart failure. Then negative cornucopia agents decrease heart rate. So again, parasympathetic innervation. So this is releasing that acetylcholine that binds to those into muscarinic receptors, which are potassium channels. So potassium exits the cell. Remember, this is causing that relaxation. So that loss of positive ions can cause hyperpolarization, and it takes longer period of time for those nodal cells to reach threshold. Remember, back we were talking about those action potentials and that potassium being released to bring it back to that resting membrane potential. Also, beta blocker drugs interfere with the binding of norepinephrine and epinephrine. And this is what we're using whenever we talk about treating high blood pressure. So for whatever reason, with high blood pressure, we've got that increase of that, we're getting an increase in pressure building up in those ventricles. So this allows, and we're going to have an increase in heart rate. So this is going to help with those beta blockers are now going to bind and block. It's doing exactly what it's saying. It's blocking that norepinephrine and epinephrine, and that's going to lower our blood pressure. So baroreceptors. Remember, we said baroreceptors are going to respond to changes in regard of stretching. So this is going to respond to changes in stretching of those blood vessels. Chemoreceptors, chemochemical. This is going to respond in changes in our carbon dioxide and hydrogen ions. Again, stroke volume, we said, is that volume of blood that's pumped from a single ventricle during that ventricular contraction.